Today I will be sharing a few more resources like how you can perform docking online or and few more resources so that it helps you in docking analysis part. And already we have done with docking with Autodoc Vina, and basic concepts and the methodologies will be you have used in Autodoc Vina will be same for other software or other approaches. Means you have to select a receptor, and maybe there might be pre-processing, and then they will perform, means the software will perform docking, and then it will perform scoring and give you some score as a result, and also the conformers or different suggestions that how uh, those two entities are going to interact. So all those things will be common to most of the packages and softwares. So you can utilize that methodology to use any software. Just that interface could be different, like you might have to use it through GUI, means graphical user interface, or command line like you have done with Autodoc Vina, or a web interface, means from website. So next also we have done analysis part, and that analysis will be common to other packages as well. So today uh, now you need to find out I want to share that how you can find out properties of a ligand. Means you need to find out what are the charges on ligand, or what is the composition of atom of composition of atoms in a ligand. So and even molecular weight matters, because all these properties are related to, means like an id to check if the drug you are predicting or the ligand you are going to dock is ideal or not, or means that also depends, tells you if your ligand is good for the for as a drug or not, or how you can interpret the properties of ligand to be good for your studies. So all those things are required. For that I would suggest you to uh, go for Swiss Param website. So this is another site, but there is a website Swiss ADME. It tells you the physiochemical or physiological properties of any drug. So if you want to know about the properties of any molecule, you can use this interface to get the properties. Like if I want to find out the properties of a drug or a chemical molecule, I need to find out the input file. Like here you can draw your molecule using Marvin JS. Means it's a web interface, so you can draw your molecule here, or you can add Smiles file here. So earlier you have already done with conversion of file formats from one format to another format, like you have done conversion of file from PDB to PDBQT, or mol to PDB, or SDF to PDB. Similarly, Smiles file format is also have been discussed, and we can convert Smile file, a file ligand structure to Smiles file. So for that either you can convert, or you can directly find out on the website. Like here I am going to share, open the PubChem, and select some random drug, and I might get Smiles file, Smile property from here. So like I am taking EGFR, and if I go to EGFR here, GFR inhibitor, I will be getting Smiles here. There are differences, like you will be getting Inchi key, Inchi key, or other type of identification. But here you need Smiles for Swiss ADME. So I will copy it. copy, paste, and then run, and it will take some time to process it and give you results. Fine. So here you will be getting most of the properties of the drug, like if like solubility of the drug or other properties which might required for the which might be required for the analysis, means processing of your drug in your body, or pharmacokinetic or thermodynamic studies of your drug. So you will be getting like solubility type of thing. So this will be helpful in designing your in vitro experiment, where you will be dissolving your drug in some solvent, like some drugs are for polar or non-polar, so accordingly you have to change your solvent molecule. So these things can be these properties will be helpful to you to know how you can prepare your drug for treatment in vitro, or even suggest your possible solvents which where you can dissolve it. So these things means you should go through this for your drug, and you will be getting most of the things as you are already aware of in biology or and sorry biology or cell signaling type of things. So these things will be easier for you to understand. And next we are going to find some online docking servers where you can perform protein docking, because it protein docking or other type of things where heavy computational resources are required and you might not be able to find those. So for that, first online web server in our list is PatchDoc, which performs PatchDoc server. So it is also available as offline tool. You can install it on your system and use that, but also it provides you online service. So you can also use download option and download it in your system to perform multiple dockings or automate it. But here it is giving you a simple interface. You need to provide your receptor molecule, or alternatively you can provide it a chain id, sorry, pdb id, and it will directly download from pdb and provide give you it as a docking result. So here it is. And next you can use ligand, or also click on this choose file to select your molecule. So you can also upload molecule and provide it some identifier uh, from here. Next you need to provide your email address so that it returns you the results in your email when done, because most of the online server queue you means put your process in queue and give result after 1 day, or sometimes it may take 7 days or 10 days. Similarly you will be seeing that you are not able to perform molecular dynamics on your local system, because molecular dynamics also takes time. So there will be online servers which might be taking 5 to 7 days to perform the docking for your study. But on your system it might take 1 year or 2 year, means it's very heavy, it requires very heavy computational resources. So like that this server help you to perform docking online. And here one more thing is there, that you can perform docking, means currently we were doing receptor and ligand. So here we had ligand, we were not able to dock protein and protein. So here ligand can be another protein, like here one example is given, that there is a pdb where ab chain is a receptor and its I chain is its ligand. So like that you can switch from protein ligand to protein docking in this web server. So it will be helping you for that as well. Next is your protein docking is also covered in this, or you can download that patchdoc or any other software you find through your literature research or any other research paper, and you can perform protein docking. Actually protein docking is important to help you understand how two proteins are going to interact in an environment, and maybe two proteins come together and form complexes that might be useful for your studies. uh, so that you can do from here. Next is that sometimes we might not be having protein structure determined computationally, so that protein structure can be found, uh, protein structure can be found on UniProt website or other servers, but means mostly all the structure which have not been determined through experiments, that are deposited on UniProt from AlphaFold. Let me show you it once. This, okay. So UniProt, here we can find some most of the means all the protein sequences which have been determined from sequencing or proteome have been deposited on UniProt KBit by AlphaFold. Means there are around 80,000 thousand structures, out of which 50,000 structures maybe have been derived through experimental processes, but still 30,000 or some critical structures are remained unpredicted. So that has been helped by AlphaFold, which is Google's DeepMind artificial neural network to provide you structures. So if uh, so since it has deposited all the structures, so we can find AlphaFold predicted structure for any of the protein molecule. So let us take example of IR alpha. It's some interleukin receptor, IL-27 receptor, IL-27 receptor. Actually, it's so it will be giving results from all the species in like mouse, human. So if you are studying related to humans or in mouse, select accordingly. I am going to choose human here, and I have say taken IL-27 alpha subunit. So for the structure I will navigate through this sidebar here, 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 and from here I will be navigating through this structure option. So here I will be getting the structure available for this particular protein. So it has only structure from AlphaFold for now, but if I go for other molecule, I will be getting experimental structures, uh, or there may be no structure at all, like in PDB only experimentally derived structures are available. So like that we due to help of AlphaFold we will be getting most of the structure. Now next thing is that we might not be having uh, a mutated structure, because we as we know that if any amino acid is going to be replaced in a structure, it is going to distort the structure. Like as a result we have seen some disease, already studied diseases like sickle cell anemia. So for to get mutated structure we need to create a structure from scratch, like we need to take a sequence, modify the residues which are going to be altered due to some mutation, and then get the structure again. That and in that case that structure not going to be may not be similar to this, and also it affects the biological process or biological features. So for to get the structure from altered sequence, we can have use AlphaFold. We can use AlphaFold to Rosetta or Modeler, and there are other paid software which can be used to get modified or mutated structures. So here we can use our Chimera X to perform this AlphaFold based prediction of structure, and there are other you to understand other software you need to go through those software and download educational version and perform analysis yourself, because for now AlphaFold prediction is far better than other already available software. So here you can directly use it, use AlphaFold using your uh, Chimera X as well. So in Chimera X I will just show you where you can find it. And online tutorial can also be find using Chimera X AlphaFold. So here you will be directly getting option for AlphaFold using some command AlphaFold, like that you will be getting options. Or you can alternatively select where uh, I am waiting for Chimera to be open so that I can directly show you that. Okay, so here if I am having some sequence, let me get some sequence. Okay, let's take it IR AlphaFold or any other P53. Sorry, I am taking example of P53. So here I am getting P53 antigen for human. Here I can also get sequence and other details here. So I'm directly navigating to sequence. This is the sequence of P53 which is not mutated, and if through literature research we might be able to find the mutated part, and we can accordingly mutate it. So like this, if there is sequence, I can if there is some mutation, like from D it is going to be other some W or other residue of protein, so we can alter it directly through sequence, and use this sequence for the prediction of our new structure. And also you can see that already AlphaFold determined structures and other details are present here. So I have gone to structure, and here there are multiple sources, like you have already seen that PDB structures are already already present, and also along with other structure predictions there is AlphaFold as well. So there are PDB and AlphaFold. So AlphaFold has also predicted here, and PDB predicted structures are already there. There are so many structures for on PDB for this particular P53 protein. So I am going here, tools, structure, structure prediction, AlphaFold, and there you can also, if you have access to modeler, you can use modeler as well here. So if I go here AlphaFold, here I can directly either fetch it or use sequence here to predict it. So, and I am just going to modify one residue, like I am changing M to A, or I will change some middle residue like P to D here. Or you can also predict some protein structure for yourself, like if you want to know how a multi uh, a chain with multiple glycines look like to prove your concepts from your theory. So you can do that also, like if I want to check how many if I have chains of only glycines, so how the structure going to be predicted, so I can do that as well here, or I can provide my sequence as well. So you can do those things, those do those things. So here you can see the instructions, like what is going to perform. If it finds some sequence of your match, it will return the result, or it will prompt you to for the predictions. So it has not found any structure for it, so I need to use predict option, and it will take me to some other some website which is provided by Google for running. It is called Google Collaboratory Notebook, and you need to log in here and provide your sequence. So I will log in here. Oh, I'm just logging it on back of it. Get the verification code here. 822705. So I have logged in here, and I will be directly taken to AlphaFold, and the look and feel of AlphaFold will be something like this, and I can directly give it my structure, or it can do something like this. And going further, it will do all type of things, like it will be downloading software, installing software, and then run the sequence prediction. Similarly, I can use the mutated structure, mutated sequence here, and get the prediction result. So like that you can predict structure, and after prediction it will come direct to a Chimera X. I am giving it time to run it. So next is that if you want to perform multiple dockings, like here we already know that for docking we need to get receptor, ligand, that may be anything, and with these both are going for the docking point. Docking, and then we are going to analyze it. So if we have 5 to 10 single receptor, we are going to follow this approach. But what if we have multiple receptor or multiple ligand? So for that we need to get some software which can take multiple receptors, multiple or single receptor, or multiple or single ligand, to perform the docking. Because for docking, we need to do the configuration type of things, and then perform docking one by one. If you have 10 receptors and 10 ligands, then it is going to form 100 complex. And if you are taking 10 minutes for single molecule docking, single complex docking, for 100 molecules it is going to 1000 minutes, and that is roughly going to be 5 what how many hours. So it is roughly going to take 1 or 2 hours continuously. So you can't that will be tedious, and if you have to reperform that to confirm the results, that is going to take around 4 hours. So we need to use some other software which can do these things for you, like PyRX, which is already optimized for Vina, or you can use FlexX, which is paid software, and other there are other softwares which may help you to perform metal complex docking of complexes with metals. Because I have already told you that you need to go through the software, its capabilities, its drawbacks, to select it for your study. And so like, so Vina is very basic, and it will be for advanced docking you need to switch to other softwares. So it will be taking some time. Actually this is taking more time because I am not using a paid version, GPU paid GPU version. So it will take some time, then it will give the result. Actually what you will get, that if your sequence is done here, sequence. Okay, so if you provided the sequence and after the prediction, it will if you click on the download, it will download the results, and the Chimera will open it here, and you will be seeing it like other results as other results. So let us wait. Anyone has any question or any problem related? So meanwhile it is running, uh, you can ask.
सर
Yes.
बेसिकली हम ग्लाइसिन हमें ऐड करना है, कोई भी प्रोटीन या अमीनो एसिड का सीक्वेंस अगर एग्जिस्टिंग प्रोटीन स्ट्रक्चर में ऐड करना है और प्रिडिक्ट करना है कि हमारा मॉलिक्यूल उसको बाइंड कर रहा है, बेसिकली लाइगेंड और ड्रग बाइंड कर रहा है नहीं कर रहा,
हाँ.
उसको हम इसके थ्रू कर सकते हैं, जो भी ये यूनिपोर्ट जो आप बता रहे हैं.
Yes, ये गूगल कोलैब, सॉरी, अल्फा फोल्ड.
Yes,
Actually,
sir.
if take example of, suppose uh, there are residues. Suppose some, this is some structure, some sequence, and there is some mutation in some part, like these two are going to change to some other residues. So if it is unmutated, it is going to require some particular 3D conformation, but for after mutation it might give some different structure. So like earlier it might be forming some helical structure, when but when there is mutation at some part here, like this, so it is
Mm,
going to change its conformation, and this alpha helix might be changing. So there
mm,
might be two resulting alpha helix, and if a molecule is going to was binding naturally here earlier, but after changing the 3D conformation, that molecule might not able to bind it, or might bind it permanently. Means based on the circumstances, the results might be different. So this altered structure will not be available, may not be available through PDB, so you will have to predict it for the
yeah.
docking.
Yes, sir. Like we have to create that PDP, new PDP ID,
Huh.
and then we can like dock on that PDP ID.
Yes, I guess it is going to take more time, maybe. Should I continue, or should we wait, or okay? It is okay, okay. It is going to take some 4 or 5 minutes, maybe.
Yes, sir.
Tesla T4 graphic processor यूज कर रहा है ये. Okay, you if you means follow this simple steps, you will be getting same result, because it's standard protocol type of thing. So you will be getting same results, like I will be showing you. So I would suggest you to perform yourself, and if you get some problem like anything, you should use screenshot to note that down. And also I will suggest you to take notes of for these things. Hello, am I muted?
No, sir.
No, no, okay, 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 okay. I thought I am muted. Okay, so
Mm.
you can means follow these simple things, and I would suggest you to perform this assignment properly, because it is going to help you understand most of the things. Repeat it, if you repeat same process again and again, and you people have invested almost 20 hours plus 10 hours. So in 30 hours you have learned the docking, and hopefully you are in better condition than wasting your time, maybe most of you.
No, sir. Wasting.
No, 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 actually if you won't have done this, you might have wasted time somewhere else, like undirected, or because I have also wasted time for to learn the docking myself. So my time was also wasted for a long time. So perform this assignment. And anyone has any questions? Shruti, Bhagisri, Vasu, you have any other question?
सर समझ में तो आ गया है, वैसे अभी ये वापस से देखना पड़ेगा सारे साइट पर. अपने एडीएमए ई समझाया है, एडीएमए के थ्रू भी हमें ड्रग. एडीएमए बेसिकली वही है ना, एड्सॉर्प्शन डिसॉर्प्शन, एड्सॉर्प्शन
Exactly,
डिस्ट्रीब्यूशन मेटाबॉलिज्म एंड एक्सक्रीशन का में
exactly,
ड्रग मॉलिक्यूल का बेसिक आइडिया लग जाएगा, जिसे हम लोग
exactly.
फॉर्मुलेशन में भी यूज कर सकते हैं.
हाँ, 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 फार्माकाइनेटिक्स फार्माडायनेमिक्स स्टडीज हम करने में इजी रहेगा. फिर
अच्छा.
ड्रग की प्रॉपर्टी और ड्रग लाइक प्रॉपर्टीज़ के लिए अदर वेबसाइट है, तो वो भी हम लोग लिस्ट आउट करेंगे. आई हैव जस्ट टोल्ड यू लिस्ट ऑफ द वेबसाइट्स विच यू कैन यूज.
ओके सर. सर अब अगर किसी को नहीं पूछा तो फिर मैं अपना एक बार डाउट पूछ लेता हूँ.
श्योर, श्योर.
बाकी आई थिंक
Okay, so I think for now docking is completed. We can go for molecular dynamics, because since we are going to we will be doing molecular dynamics, we will be needing molecular docking repeatedly. So one day I will follow complete process so that you understand docking
सर
clearly.
डायनेमिक स्टार्ट करने से पहले मेरे ख्याल से आपको ना इंडिविजुअली सबसे ना एक मतलब एक क्लास में मतलब जैसे असाइनमेंट टाइप हो या एक इंडिविजुअल क्लास में
हम्म,
मतलब हर कोई मतलब एक क्लास में कैसे दो दो तीन तीन बच्चे मॉलिक्यूलर मॉलिक्यूलर डॉकिंग समझा सकते हैं मतलब कैसे काइमेरेक्स में हम कैसे मतलब पूरा विजुअलाइजेशन के साथ लेकर टिल मॉलिक्यूलर डॉकिंग स्कोर आने तक का पूरा मतलब जब तक कोई इंडिविजुअली करके दिखाएगा नहीं ना
हाँ हाँ।
क्लास में तो मुझे लगता है वो ज्यादा यूजफुल होगा मतलब अगर किसी को समझ में नहीं भी आएगा तो वो भी समझ लेगा उस हिसाब से।
Okay, okay. So what I will do, I will create groups, and
हम्म
then one means 1 week, like 2 days, we will be
हम्म
doing this. So it is almost going to take 10 to 20 minutes per single student, and they
हाँ
will
सर
be able to perform and perform the docking. Okay, so I will
हाँ
do
सारे
that.
सबको मतलब ऐसे ये होगा कि सबको सबको क्या मतलब जिससे नहीं आ रहा होगा उससे समझ में भी आ जाएगा और उससे मतलब वापस से रिपीट भी हो जाएगा।
Yes, okay, I will do that, because most of the
और
people are not confident, I guess.
नहीं सब लोग कर ही लेंगे सर दिक्कत जो जैसे मधुलिका हो गई और वो गोपीनाथ हो गया एक दो और स्टूडेंट संजय हो गया ये सब तो कर ही सकते तो करते भी है और
They
दिखाते भी है तो
yeah,
मुझे नहीं लगता।
they are able to perform. Means there are other students who just watch and not feel confident maybe. Okay, so
हाँ तो
I will do that. I will schedule that.
हम्म, आप वो ना ग्रुप ग्रुप ये मतलब शेड्यूल बना लेना मतलब किसका कब है सो
Uh-huh.
दैट वो उस हिसाब से फिर वो रेडी रहे।
Okay, so I will share that schedule in the group.
हाँ
Any other question?
और सर ये अभी आप मल्टीपल डॉकिंग अभी बताओगे ना कि मल्टीपल डॉकिंग
हाँ,
किस
वो
मतलब
परफॉर्म
किस
कर।
तरीके से करते हैं।
यस यस, बिकॉज एवरीथिंग एट मीन सारी चीज़ें एक साथ बताने से तो क्या रहेगा कि कंफ्यूजन भी होगा मे बी, और मुझे भी लेके प्रिपेयर करने के लिए टाइम चाहिए होता है ना, तो
यस सर यस
I
सर।
will find optimal resources so that multiple docking thing is easy for you. I will start with basics of molecular dynamics and keep that thing docking thing parallel. Like in middle, I will be giving assignment related to docking. So if earlier you might be taking more time, like 1 hour, 2 hour for to perform docking or practice it, but over the time you will be spending only 5 to 10 minutes to perform docking and analysis and preparation and all those things for from docking point of view, because the molecular dynamics is going to take more time than expected.
यस सर।
Okay, so from here I will start with the molecular dynamics. So because I wanted to dedicate 2 months for molecular docking and the remaining 4 months for molecular dynamics. So but it has been means kind of disbalanced.
ऐसा कुछ भी नहीं है सर वो तो करने के बाद मतलब हम जैसे शेड्यूल बनाते हैं वैसे होता नहीं है लेकिन फिर भी स्टिल मतलब कर तो रहे ट्राई।
हाँ हाँ, वही जस्ट आई वांटेड दैट कि कीप ट्राइंग। अगर फेल हो रहे हो तो रिजल्ट, कम से कम कुछ तो रिजल्ट बताओ जो फेल होने के लिए भी आ रहे हो तो भी चलेगा।
हाँ सर।
ठीक है, तो कोई और क्वेश्चन है तुम्हारे पास अभी?
सर मेरे पास और कोई क्वेश्चन अभी है नहीं हाँ एक क्वेश्चन और है
हम्म
जैसे कि अभी मॉलिक्यूलर डॉकिंग में मतलब सर ने मुझे बोला है मेरा जो गाइड है
हम्म
उन्होंने बोला है कि आप वे टू ड्रग में जाकर अपना मॉलिक्यूल रखो जैसे कि मेरा मॉलिक्यूल वेलेरिक एसिड है ठीक है
हम्म
सर। तो सर बोले कि उसको आप वे टू ड्रग में जाकर उसको वहां पर मतलब पेस्ट करके या उसको अपलोड करके वहां पर देखो कि कौन कौन से इसके टारगेट है तो सपोज करो एक मॉलिक्यूल के अंदर मतलब 50 टारगेट है ये पाथवे जिससे वो बता रहा है।
ओके, सो दैट
ठीक है
मीन्स प्रेडिक्टेड रिसेप्टर्स और प्रेडिक्टेड टारगेट्स, राइट?
हाँ सर। तो सर अगर सपोज करो मेरे पास 50 मॉलिक्यूल्स है तो मैं 50 की 50 सबको मैं उसमें जाके चेक करूं और उस फाइल को सेव करूं द फर्स्ट 20 फाइल को सेव करूं कि ये मेरे प्रिडिक्टेड रिसेप्टर्स बन सकते हैं।
मैं सजेस्ट क्या करूँगा कि हम लोग इसको बाद मैं स्क्रीन शेयर करना तुम मेरे साथ, और
अभी
देन
कर ये आप अभी बिजी हो आज।
बिजी तो नहीं हूँ, ठीक है शेयर कर लो।
नहीं, अगर आप बिजी हो तो फिर नेक्स्ट टाइम भी।
अगर नहीं मीन्स जस्ट 5-10 सेकेंड 5 मिनट में होता है तो ठीक, इट्स ओके ना।
अच्छा ठीक है। और सर वो ना अभी भी रिजल्ट नहीं आ रहा, अभी पता नहीं उसमें रिसेप्टार में भी गड़बड़ बता रहा है, मुझे नहीं पता क्या दिक्कत हो रहा है, समझ ही नहीं आ
प्रिपेयर
रहा मुझे।
रिसेप्टर फिर से प्रिपेयर करो, डॉकिंग में अगर जो भी एरर्स आ रही है ना, उसको
हम्म
प्रॉपरली
हम्म,
विजुअलाइज करो।
ठीक है। अभी रिसेप्टार पे वो ये बोलना चाह रहा है, प्लीज ना पार्स एरर ऑन लाइन 2 इन
दैट
फाइल
मीन्स
आर।
that means your file is not properly prepared. You need to prepare it again, or tell me the send the pdb id in the group, so anyone else tries that and gets same error, then we can do something. Means I will try, I will try to find other solution for that.
मैंने सोचा कि शायद हेट्रोएटम में भी शायद होगा, बट मैंने हेट्रोएटम भी सर्च करके
No,
देखा, एच
heteroatom doesn't
हेट
matter. Heteroatom and atoms doesn't matter for the talking. Means there might be something which might not be understood by Vina software, or maybe new, so that might be giving you error.
सर मैं फिर से ट्राई करता हूँ अभी। मैं आपको ये बता रहा था, गो फॉर प्रेडिक्शन।